A warm welcome to all. Today we discuss about chromatography technique. It is very important to analyze the component technique in any solution. I am S.Veeralakshmi, Assistant Professor of Department of Microbiology, the Standard Fireworks, Rajatnam College for Women. In this video, we learn the terms and principles and applications of chromatography. Principles of chromatography. The chromatography is an analytical technique commonly used for separating a mixture of chemical substance into its individual components so that the individual components can be thoroughly analyzed. In this technique used for the separation and purification and testing of any compounds, the term chromatography is derived from Greek word. The chroma means color and the graphein means to write. The chromatography was first devised at the University of Kazan by Italian-born Russian scientist Mikhail Twist in 19,000. He developed the technique and coined the term chromatography in the first decade of the 20th century, primarily for the separation of plant pigments such as chlorophyll, carotenes and xanthophylls by using chromatography techniques. So he called as father of chromatography. The terms used in chromatography. The first, chromatograph, an instrument used to perform chromatography which separates mixtures into individual components. The next, eluent, the solvent or mobile phase that carries the analytes through the stationary phase in chromatography. Next, the elution, the process of passing the mobile phase through the stationary phase causing the separation of analytes. The stationary phase, the phase is chromatography that remains fixed in place while the mobile phase moves through it. It is usually a solid or a viscous liquid. And then mobile phase, the phase that moves in definite direction carrying the analytes with it. It can be a liquid or gas in nature. Retention time, the time it takes for an analyte to pass through the chromatography system from the point of injection to detection. Next one is analyte, the substance or component being analyzed and separated in chromatography. Chromatogram, a visual output of the chromatography process showing the separated components as peak on a graph. And then last, solvent, the liquid in which the analytes are dissolved to form the mobile phase. And that we'll see about principle. Chromatography is based on the principle where molecules and mixture applied onto the surface or into the solid and fluid. Stationary phase is known as stable phase, is separating from each other while moving with the aid of a mobile phase. The factor effective on this separation process include molecular characteristics related to adsorption, it's a liquid and solid nature, partition, liquid and solid nature, and affinity or differences among their molecular weights. Because of these differences, some components of the mixture stay longer in the stationary phase and they move slowly in the chromatography system, while others pass rapidly into the mobile phase and leave the system faster. There are three components that form the basis of chromatography technique. First one is stationary phase. It is very important. This phase is always composed of a solid phase or a layer of a liquid absorbed on the surface solid support. And the next, mobile phase. This phase is always composed of liquid or a gaseous component. And the next, separated molecule. The type of interaction between stationary and the mobile phase and substance contained in the mixture is the basic components effective on the separation of molecules from each other. And then next we learn types of chromatography. There are many types of chromatographies available. The first, thin layer chromatography. It is the thin layer chromatography and high performance liquid chromatography, gas chromatography, column chromatography, paper chromatography and ion exchange chromatography, and then last, affinity chromatography. These all types are used for the chemical components analyzing method. And then final, we see application. A wide range of applications is present in the chromatography technique. The first, pharmaceutical. It is used to analyzing during drug purity and composition, and the environmental science we detect the pollutants in air, water and soil samples, and then food industries ensuring the food safety and quality, and forensic science we analyze the substances in criminal investigations in murder cases or another cases. Biochemistry and molecular biology to purify the nucleic acid, proteins and other biomolecules. These are the general principles about the chromatography. Thanks for watching this video. We meet another video.